There are relatively few drugs available to treat Mycobacterium tuberculosis, so resistance to any drug is a concern. This video article describes a procedure for testing the antibiotic resistance of M. tuberculosis to 12 different microbial drugs simultaneously. Thus, the ability to rapidly detect antimicrobial resistance of M. tuberculosis is very important in the effort to control the increase of resistant strains. Here, to test antibiotic susceptibility, M. tuberculosis is first grown on solid or in broth media. A bacterial suspension, the inoculum, is prepared and added to a microtiter plate containing different concentrations of 12 drugs, including first-line and second-line drugs. Following incubation, the extent of growth in each well is determined either manually using a mirror box or using a semi-automated plate reader. The resulting data indicate the minimum inhibitory concentration for each drug tested, and show which antibiotics the tested strain of TB is resistant to. These results aid in choosing the appropriate drug for treatment of TB.
Hi, my name is Nancy Wenjanek, and I'm director of the Mycobacteriology Laboratory at the Mayo Clinic in Rochester, Minnesota. Today we're going to describe a method for doing antibiotic susceptibility testing for Mycobacterium tuberculosis. The main advantage of this technique over existing methods, like macrobroth dilution, is that an MIC is determined for each drug and 12 antibiotics are tested concurrently. Demonstrating the procedure will be Kurt Jude, a quality specialist in my laboratory.
Working on a laboratory bench, prepare for this procedure by labeling a blood agar plate, 3 Middlebrook 7H10 plates, a saline tween glass bead tube, and a microtiter plate with the patient's name and specimen identification number. Additionally, label the blood agar and 7H10 plates as purity check. Label one of these 7H10 plates as 1:1 colony count, and the other as 1:50 colony count. Then place the tray in the pass-through. Next, prepare to work in the Level 3 Biosafety BSL-3 facility by putting on a solid front gown, head cover, gloves, shoe covers, and a respirator. Here, a PAPR respirator is used. Then retrieve the tray from the pass-through. Next, in the BSL-3 facility, disinfect the surface of the biological safety cabinet. Then place a paper towel soaked with disinfectant inside the hood, approximately 6 inches from the air vent panel. Next to the paper towel, place inoculating loops, pipetters and tips, an appropriate waste disposal container, a nephelometer, and a vortexer. Once the hood is prepared, place the organism on the paper towel, along with a rack with a McFarland 0.5 standard, a 7H9 broth with OADC, and a saline bead tube. This setup is done from solid medium. Prepare inoculum from a solid medium culture from a patient's specimen. Use a sterile loop to scrape the colonies and transfer them into the saline tween glass bead tube. Vortex the suspension for 30 to 60 seconds. Visually inspect the sample, comparing it to the McFarland standard. The amount transferred should be equal to or greater than the standard. If not, add more of the bacterial sample and vortex again. Once the McFarland standard is matched, allow the inoculum to settle for 15 minutes to allow larger clumps of bacteria to settle. Next, use a nephelometer to measure the density of the suspended bacteria. To zero the nephelometer, first place a blank saline bead tween tube in the nephelometer. Press the calibrate button to zero the instrument. Then place the McFarland 0.5 standard in the nephelometer and press the calibrate button again. Once the instrument is calibrated, remove the McFarland standard and insert the inoculum. The inoculum reading should be equal to the McFarland standard reading. If the reading is too low, remove the tube and transfer more bacteria from the plate. Then vortex and read the sample again. If the reading is too high, add saline tween to the tube. Vortex, then read the sample again. Continue adjusting the inoculum as needed until the concentration of the bacteria matches the McFarland standard. Next, use a 200 microliter pipetter and a long aerosol-resistant pipette tip to remove 100 microliters of inoculum from the top third of the tube to avoid any clumps that are settling to the bottom. Inoculate the Middlebrook 7H9 broth. Slowly vortex the inoculated broth for 20 seconds. To plate the inoculum, begin by removing the microtiter plate from the manufacturer's package. If the desiccant is blue, or if the foil package is damaged, do not use it. The plate already contains the 12 antimicrobials to be tested in each well, including the most effective or first-line drugs and second-line drugs. Carefully pour the 7H9 broth inoculum into a trough, taking care not to generate aerosols. Then place the microtiter plate with the label facing out, as shown here, and use a 12-channel pipetter to add 100 microliters of the inoculum to each well. Next, prepare plates for performing colony counts. First, prepare an undiluted plate by transferring a 1 microliter loop of sample from the positive control well to the 7H10 plate labeled 1:1. Then use the loop to streak the plate, as shown here. Next, to prepare a 1:50 dilution, transfer a 1 microliter loop from the positive control well and swirl it into the tube with 50 microliters of water. Streak 1 microliter of this dilution on the plate labeled 1:50. Inoculate the purity plates by pipetting 50 microliters of the inoculum from the trough onto appropriately labeled blood agar and 7H10 plates and streak for isolation. Then stack another trough on top of the inoculated trough and gently place them into the discard container. Place an adhesive seal on the microtiter plate. Then, using the white backing, press firmly on each well to assure adequate sealing, ensuring that there are no creases. Disinfect the microtiter plate by wiping it with a paper towel soaked with a tuberculosidal disinfectant. Then place the microtiter plate in a plastic bag and close it with a heat seal. Finally, disinfect all materials, including the nephelometer, vortexer, plates, and tubes, by wiping them with a tuberculosidal disinfectant. Here, ProSpray is used. After enough time has passed to ensure disinfection, 15 minutes for ProSpray, the plates are taped with Scotch tape, placed in plastic bags, and sealed. Supplies are removed from the biosafety cabinet and placed in the pass-through. The discard red bag is closed and sealed with a twist tie. At this point, remove the gown, booties, and gloves. After proceeding to the anteroom and washing hands, remove the respirator and disinfect and store it. After washing hands again, put on a fresh gown and leave the BSL-3 suite. Next, the discard container is autoclaved. Following this, incubate the microtiter plate and the 7H10 and blood agar plates right side up in the 35 to 37 degrees Celsius incubator with 5 to 10% CO2 in the BSL-2 laboratory. This is acceptable because the plate is well sealed and in a plastic bag. Mycobacterium tuberculosis generation time is approximately 12 to 24 hours, so it will be at least a week before growth can be seen as the formation of a cell pellet in the bottom of the control well. On day 10, perform a manual mirror read by placing the sealed AST microtiter plate onto the platform of the mirror box. Use a desk lamp to aid in visualizing the growth. Adjust as needed to get the best contrast. Check the growth control wells, in this case H11 and H12, by looking at the mirror. If there is growth, the well will appear cloudy or appear to have sediment, as shown here. If there is no growth, as shown in this example, the well will appear to be clear. In this case, place the plate back in the incubator and continue incubating for up to 21 days. For each condition, compare the growth to the control well. Then for each drug, record the first dilution that has no growth. This is the minimal inhibitory concentration, or MIC, for the drug. To examine results using an automated plate reader, such as the Vision, begin by opening the MycoTB AST plate software and logging in. Place the sealed plate with the barcode facing out, as shown here. Growth appears as turbidity, or as a deposit of cells at the bottom of a well, on the computer screen. First, examine the growth controls. As before, if this is negative for growth, place the plate back in the incubator for up to 21 days. If this is positive, record the results by touching the first well that does not show growth on the screen for each drug. In some cases, bacteria sediment or drugs precipitate to the bottom of the well. This phenomenon, known as trailing, is indicated by several wells with the same size pellet and is not actually considered positive growth. Next, the 7H10 and blood agar plates are examined to determine bacterial concentrations and assess purity. The blood agar plate should show no growth after 48 hours. Growth on the blood agar plate indicates contaminations with rapidly growing bacteria. If there is growth, the microtiter test must be repeated. The 7H10 purity plate should have growth of one organism type. M. tuberculosis should appear buff-colored, wrinkly, and rough. If contaminated, the microtiter test needs to be repeated from a pure culture. To assess bacterial concentration, count the colonies on the 7H10 plate. Then, using the table in the accompanying written document, calculate colony counts. Mycobacterium tuberculosis was grown from a patient specimen and antibiotic resistance was assessed, as described in this video. In the plate shown here, 12 different drugs are present in increasing concentrations, with the highest concentrations at the top and the lowest concentrations at the bottom. The minimal inhibitory concentration, or MIC, of the drug was indicated by the lowest concentration of the drug that did not show any growth and is circled in the image shown here. Using standardized breakpoints established by the Clinical and Laboratory Standards Institute, the data shown here suggests that this strain of TB will be resistant to isoniazid and rifampin in vitro. The high MICs seen in several other drugs, including ofloxacin, moxifloxacin, streptomycin, and rifabutin, suggest the resistance of the M. tuberculosis strain to these agents. The MIC results should be used by an infectious disease specialist experienced in the treatment of tuberculosis to develop an individualized treatment plan.
Once mastered, this technique takes about 40 minutes to set up one isolate. While attempting this procedure, it's important to remember that this is a respiratory pathogen and one should not generate aerosols. After watching this video, you should have a good understanding of how to set up Mycobacterium tuberculosis antimicrobial susceptibility testing using the microbroth dilution method. Don't forget that working with Mycobacterium tuberculosis can be extremely hazardous, and precautions such as respiratory protection should always be taken where required.